So welcome everyone to our tutorial about epigenomics data analysis, part of the ECCB 2020. I'm very excited that so many people joining us today to learn how to analyze epigenomics data. This tutorial is, as you hopefully have seen, uses a Docker. So in fact, all the data and the software that you're going to use is part of a Docker image, and which is explained in the documentation that belongs to this tutorial. So if you haven't done so, install Docker and use this command, which is also listed in the documentation for getting that image, because everything will happen in that image. Yeah? And so you can actually, because there's going to be a talk now, if you haven't done so, you can still download it. It's something like 20 GB, so it's big. Yes, and so that is very essential. I hope that everyone is able to do this. We've been debugging this and tested this on all systems, you know, Linux, Mac, and Windows, and we've always made it work. So hopefully there's no problems with this. But let's see. Hello? Do you comment? No. So.
Marcel, you're on mute.
So this is the documentation. This is the link, yeah, which just describes everything that's going to happen in this tutorial. Here's how to install all of this with all the details. And then we're going to have different practical sessions, which you're going to go through after you heard some details about analysis. And so the exact schedule is this here. I think you've all seen it. So we will have a first part, a lecture about methods and algorithms for epigenome analysis in the context of open chromatin data mostly. So peak and footprint calling, and then transcription factor, motif analysis in those regions, and basically using the ATAC data to trying to understand differences in transcription factor binding. Following the first practical, where you're going to actually analyze real data, using software that's part of the Docker image. And then we're gonna, in the second lecture, talk about approaches for identifying target genes and regulatory elements, and also prioritizing PF regulation with gene expression data. And this is then what you're gonna do in the second practical, which we've separated into two parts, so practical 2 and 3. Basically where you are gonna learn how you link regulatory elements to genes, or like footprints, for example, to genes. And then, we hope we can then discuss, results of all of those with you, and at the end wrap up. And hopefully this is very helpful to you. So the people that are involved in preparing this tutorial are from 2 different universities. The first is in Aachen. It's from the group of G Costa, who's sadly couldn't join us today, and Zhijian Li, one of his colleagues. They are basically responsible for the first part of this tutorial, the ATAC-seq analysis. And the second part is people from my group, Nina Baumgarten, Dennis Hecker. Oh, sorry, there's a typo here. This is of course not. So this is his name. So Sivarajan Karunanithi is the other person from Goethe University, and me, I'm Marcel Schulz. So we've put all the work into preparing this for you. And what are you going to do? Well, we're going to look at published data from this very paper. This is called an Atlas of Transcriptional Chromatin Accessibility and Surface Marker Changes in Human Mesoderm Development. And these authors had looked at differentiation of pluripotent stem cells, HESCs, and then basically through different days of differentiation, certain progenitor cells are derived, and they've looked at the epigenomic profile and expression profile of those. And we're going to compare in this tutorial pluripotent stem cells, HESCs, with cardiac mesoderm stem cells. So basically, stem cells from the mesoderm lineage will be derived. And so we're going to try to understand, what kind of epigenomic changes have occurred through these 3 steps of differentiation, and which transcription factors are involved in, this regulation. And so, as I mentioned, there will be 3 parts of a hands-on analysis that you're going to do, that we're going to teach you. The first part, we're going to use that data and predict sets of attack peaks and footprints, and you're going to learn what that is. And with these, we're going to see which transcription factors seem to be genome-wide changing their binding profile, potentially. And then in the second part, we're going to look at those footprints, and we're going to use that together with a set of differentially expressed genes between these 2 conditions that was measured with RNA-seq, which transcription factors seem to explain now changes in those differentially expressed genes. And then in the last part, we're going to use now transcription factors that we've found to be of potentially interesting result in these first 2 parts. We're going to look into how we can now associate potential target genes for these transcription factors. We're going to look at basically different ways how you can do this, and then discuss those results. So that at the end of the day, we hope that you'll be able to know how you can have to analyze ATAC-seq data and then integrate with gene expression data, which is a common thing, to reason about the process under study, and then also prioritize transcription factors and their target genes that are relevant. So, in terms of interaction during the tutorial, as I said, there's going to be lectures and then hands-on series. In this, I hope you had a look in this ECCB portal app, right, in this barceloeventsair.com. There's on the right side, you can file in questions, as some people have already done. And so please do so during the talks. You can also raise your hand here in Zoom, I believe, right? If you want to speak to us and have like a very specific questions also as part, maybe when we're discussing, and so we can unmute you, you can actually talk to us. And yeah, we hope that this gives us sort of like enough flexibility that we can help you out with all the problems and answer all the questions you have. Okay, so I think that's pretty much all I wanted to say. And so then I would say, let's not waste any more time and let's get started. So Zhijian Li is going to do the first part of the presentation. So could you take over?
Yeah, sure. Let me share my screen. So now, can you see my screen?
Yeah.
Okay, and hello everyone. My name is Zhijian Li, and I will be talking about method and algorithms for epigenome analysis in next 30 minutes. Specifically, I will first talk a little bit about the background and ATAC-seq protocol. Then I will show you a standard pipeline to analyze ATAC-seq data, including quality check, reads, alignments, and p-calling. Next, I will introduce you for the printing analysis and show you how we use it to study dendritic cell differentiation. Finally, I will give a brief description of practical, and after this, we will go to the practical parts where we provide you some real data. And you will try to run the pipeline to perform for the printing analysis. So this picture shows you the hematopoiesis differentiation model, where a progenitor cell such as long-term hematopoietic stem cell branches into distinct lineages. And it's known that transcription factors are controlling the cell specification. For example, the differentiation from CRP to T cell is controlled by GATA3, as you can see here. Now the question is, given any cell differentiation model, can we figure out which factors control cell specification? One computational approach will be motif searching, where we use motifs and DNA sequence to do motif matching. The motifs usually come from some public datasets. For example, here to study which factor is regulating the expression of gene X, we can perform motif searching and we can find the motif of PO1. However, this approach has several problems. The first one is the motifs are usually small and degenerate. The second one is the binding sites of transcription factors can be many kilobases distant from the gene that they regulate. Last but not the least, the public datasets are usually incomplete, meaning that only part of motifs are known. So, given this method will generate too many false positive predictions. On the other hand, as we know, chromatin accessibility plays an important role in establishing and maintaining cellular identity. So in this picture, we can see that the gene expression is controlled by chromatin accessibility. For example, if the chromatin is accessible, transcription factors can bind to the promoter and activate the gene expression. Okay, so to measure chromatin accessibility, people have developed different protocols such as DNA-seq, ATAC-seq, and MN-seq. In this tutorial, we will focus on ATAC-seq. This is how ATAC-seq works. Basically, ATAC-seq uses a hyperactive transposes called TN5 to simultaneously cleave double-strand DNA and insert sequencing adapters. Then those DNA fragments are then purified, PCR amplified, and sequenced. After alignment, a signal will be generated and the accessible chromatin regions will be defined by epigenomic. To check the quality of ATAC-seq data after alignment and picoling, we can use the fraction of reads in peaks, or we call FRIP. This metric provides an estimate of the signal versus noise in a library. By signal, we mean the reads which are from peaks, and the noise indicates the reads from background. Usually, we recommend to have a FRIP higher than 0.3, which is also used by ENCODE project. As you can see here from these plots, the ATAC-seq from the stem cell have a lower quality. We can also check the fragment size distribution because currently ATAC-seq libraries are mostly based on pair-end sequencing. So the fragments are either from nucleosome-free regions or the regions that contain nucleosomes, as shown in this picture. So the fragment size distribution provides information about nucleosome positions, as you can see here from the right-hand side. A good library should have a clear periodicity. Of course, there are several methods to do the picoling for ATAC-seq. And in a paper from Genome Biology, the authors compared different methods for ATAC-seq picoling. And here I would like to highlight two of them. One is MAX2, which was designed for ChIP-seq, but now is widely used for ATAC-seq. Another one is HMMR ATAC. This method is specifically developed for ATAC-seq by considering fragment size distribution, as I mentioned before. Besides picoling, we can also do differential picoling by finding genomic regions with changes in rate density between two conditions. However, currently, no differential peaks analysis tool have been developed for ATAC-seq. So, a straightforward approach would be to first find the candidate regions, which can be done by peak calling for each condition independently, and then we can count the number of rates in those regions, and then compare with other conditions statistically. For example, using DE-seq2. So this workflow summarizes the bioinformatics pipeline that we usually use to analyze ATAC-seq data. Next, I will talk about ATAC-seq footprinting that we think might be useful to analyze ATAC-seq data. So this picture shows you how computational footprinting works. The principle is if there is some factor binding to the DNA, and then those DNA will be protected from the TN5 cleavage. So if we generate the ATAC-seq signal per nucleotide by only counting the start size of the fragments, we will be able to find out the protected regions, which we call footprints, as shown on the bottom here. To do footprinting, we have proposed a HM-based model called HINT. So HINT will first generate normalized cleavage signals, and then we train HINT with limited supervision. Finally, we use multivariate signals to predict footprints. Here is how we evaluate the prediction of HINT. Basically, we first predict footprints using ATAC-seq and HINT, and then by combining with motif matching results, we can check if a binding site is supported by footprints or not. And then we can have the predicted cell-specific binding sites for a particular factor. For example, here we highlight the predicted binding site for CTCF. And next, we can use ChIP-seq as true labels to classify our prediction as true or false. By using this, we are able to compute the AUPR or area under precision recourse curve. And a higher value means a better prediction. Once we have predicted the cell-specific binding sites, one thing we can do is to create a ATAC-seq profile around the predicted binding site and use it to compute a transcription factor activity score by measuring the depth of ATAC-seq signal. Because we assume that if a factor has higher activity, the chromatin will be more accessible around the binding sites of this factor. So the footprints will be deeper. To improve the prediction, we also needed to consider the cleavage bias in ATAC-seq. As shown here, this motif logo represents TN5 cleavage preference, meaning that some sequences have more cuts than expected. To solve this problem, we propose to use variable order Markov model to estimate the bias and correct the bias in signal generation. This scatter plot shows the comparison of HINT with and without using bias correction. Each point here represents a transcription factor, and the x-axis is the EUPR of HINT without using bias correction, and the y-axis is HINT with bias correction. So it's clear that the prediction can be improved by using bias correction. We can also look at the average ATAC-seq signal from the predicted binding sites before and after using bias correction to see how the signal is changed. So as shown here in the right-hand side for SP1, after bias correction, the footprint becomes more clear. We can also compare the signal on a single position level. Again, by using bias correction, we are able to identify a footprint which is supported by motif and chipstick, as shown here. Since ATAC-seq provides information about the nucleosome positioning, as I mentioned, so we're also wondering if we can use it to improve the footprinting. To do so, we proposed nucleosome position decomposition, and we first classify the ATAC-seq rates as nucleosome-free rates, as you can see here, and rates with one nucleosome or rates with more than two nucleosomes by using their fragment size. And then we are able to create the signal for each of them, as you can see from the right-hand side here. On the left, we can see the evaluation result using different ways to do the decomposition. And it turns out that by decomposing the rates as nucleosome-free rates and rates with more than one nucleosomes, we can improve the prediction significantly. So finally, we propose the HINT-ATAC by combining bias correction and nucleosome decomposition. We also compare the HINT-ATAC with other competing methods. On the right-hand side, you can see the benchmarking results, and we can see that HINT-ATAC outperforms other competing methods. However, one major problem of using footprinting is that the prediction usually have a low performance for those factors with low residence time, meaning that they are poorly detected by footprint. And this problem actually exists in both ATAC-seq and DNA-seq. Next, we used ATAC-seq footprinting to study regulatory control of dendritic cell differentiation. Specifically, we want to ask which factors control dendritic cell differentiation and how is chromatin regulated. To answer this, we have been using chip-seq and iron-seq, and we were able to identify PO1 as a master regulator of CDP differentiation. Here, we would like to extend the study by measuring the open chromatin with ATAC-seq. So working together with Professor Martin Zink from UCA Aachen Cell Biology Department, we performed ATAC-seq on PDC and CDC. After processing the data, we predict the footprinting in CDC and PDC independently. And then we calculate the transcription factor activity score for more than 500 motifs. As shown in the middle, the y-axis represents the differences of TF activity between PDC and CDC. A positive value means a higher activity in PDC, and a negative value means higher activity in CDC. Here, we highlight factors with significant footprint change. So based on this analysis, we can identify PDC-specific factors, for example, TCF4, as well as CDC-specific factors like BATF3. On the right-hand side, the line plots show you the ATAC-seq signal around the binding sites of TCF4 and BATF3. So we can say that the chromatin around TCF4 binding sites is more accessible in PDC, while the chromatin around BATF3 is more accessible in CDC. By using this way, we are also able to identify some unknown factors as colored by blue. So this analysis provides a candidate list for further study. To investigate how BATF3 regulates gene expression in CDC, we here look at the two genes. For example, IRF8 and FLT3, which are known to be relevant for CDC differentiation. And from this picture, we can see that the predicted footprints, as shown in the middle, are supported by both motif and chip-seq, meaning that HINT-ATAC provides highly specific prediction. On the other hand, without using footprinting, we will have more false positives, as shown here on the bottom. So to summarize, here we briefly overviewed the basic ATAC-seq analysis pipeline, including quality check, rate alignment, and peak coding. Then we introduced the computational footprinting and explain that how we develop HINT-ATAC to predict transcription factor binding sites. Finally, we showed one example where we use HINT-ATAC to study regulatory changes of dendritic cell differentiation. So with all of this, I hope I can convince you that footprinting is helpful and is able to generate new biological insights. Next, we will do the practical session 1, where we are going to analyze ATAC-seq data from stem cell differentiation. Please be sure you have installed Docker and IGV before you start the practical session 1. So I will try to go through the practical, and I hope you can follow me to do the analysis step by step.
Lee, we got a question in the the current questions.
Okay, so we can do some QA since we have 10 minutes left. The question. So how much? Actually, we haven't checked the situation or overexpression of the data. Basically, we only check the FRIP for one experiment, and if the FRIP is higher than 0.3, as I mentioned in the talk, then we think the data is fine. So is there any other questions? Yeah, sure. The slides will be uploaded to GitHub, and it will be free to download. And we have 10 minutes left. So if you have any question, just post to the platform. And if there is no further question, I think we can start to do the practical one. So I will try to run the pipeline on my computer. And I hope you can follow me. Let me first share my screen. So Dennis, can you see my screen?
Yes.
Okay, then fine. So just be sure you have followed the installation structure to be able to set up the Docker. So in the first practical, we will perform basic analysis of the ATAC-seq, including footprint analysis and the motif matching with HINT to identify cell-specific binding sites from open chromatin data. The first step is to check the data quality. And here, since we have performed the low-level analysis, including raised alignment and the picoling, by using this pipeline from Nextflow, so you can directly go to the container data folder to check the results. And we will first check the data quality as I talked in the presentation. We will look at the FRIP. So this report was generated by the NF core ATAC-seq pipeline, and you can check the data quality. For example, go to here. So because for this data set, we have two replicates for each condition. So you can check. So here we have 4 different numbers to indicate the FRIP for each of them. And we can say that the cardiac library have a relatively good quality, but the stem cell has very low quality as the FRIP is quite low. You can also go to you can also check the fragment size distribution. Let me see. Okay, just go to the insert size, and here you can look at the fragment size distribution. I don't know why, but and next we can check the which minutes? Next, you can use IGV to visualize the ATAC-seq signals and the peaks for a particular loci. So to do so, we can use IGV, and we can import the session that we create for this dataset. Open session. Go to the container data. HV. So as you can see here, we can actually look at the ATAC-seq signal for the stem cell and the cardiac progenitor cells. So you can also just check for a particular gene. For example, here I will check the chromatin profile for gene GATA4. So I can just type GATA4 here. And then we can check the chromatin profile for GATA4. So as you can see here, the GATA4 has higher chromatin accessibility in cardiac cells, which is known. So next, we will try to run some commands to do footprinting analysis. First, we need to create a folder for the results. And because if we run the analysis by using all chromosomes, we will not be able to finish in 30 minutes. So here we only consider peaks inside chromosome 21. So we can do this whole analysis in 30 minutes. To select the peaks for only chromosome 21, you can try to run these two commands. Okay, so is everyone able to run these commands? If you have a problem, just let me know. Hi, Dennis. Is everything working fine? You can hear me, right?
Yeah, we can hear you.
Okay. Just got no feedback. And so if we have the peaks for chromosome 21, then we can try to run HINT to perform the footprinting. Just copy and paste the command here. We first create a folder for the footprinting. And then we run the HINT-ATAC to code the footprints. So these commands will usually take 2 or 3 minutes, depending on the machine that you are using. So let me see, still running. Okay, still running. So if you have a problem, or questions, let me know.
Lee, I think you don't have to have the Zoom panels open. I will tell you when you're not audible or when the screen sharing doesn't work, because we can see ourselves now.
Okay, yeah, just to be sure everything is working fine.
Yeah, I will let you know. And also, I mean, do you see the? I will tell you when there are some questions on the website itself.
Okay, so. Cool, great, thanks. So now we are waiting for the finish of the printing. Takes longer than expected. Usually it takes 2 minutes on my computer, but I don't know why today it takes a little bit longer.
Well, you know, probably your computer is busy with all the other things going on. The video.
Yeah, the video, the browser. But
because it's running this kind of virtual machine, right? So in the background.
Yeah.
Maybe can you take away the Zoom window, because you're forwarding the Zoom window in your. You put it to another monitor or make it smaller. Thanks.
Still running. So when we try to when we try to use RGD hint to do the printing, I can explain to you a little bit about the parameters that we use. So this ATAC-seq here means you are doing ATAC-seq to do for the printing, because we also support different data sets. For example, we support DNA-seq. We also support to use histone modification data. And here, parent means that the library that we are using is parent sequencing. So as I mentioned before, by using parent sequencing, we can calculate the fragment size and then use the fragment size to improve our prediction. And here means we are using the different references genome. We specify the genome version as HG38. And next is we specify the output location and the output name. These two parameters indicate the input for RGT footprinting. The first one is the BAM file, which is generated by the NF core pipeline. The second one is the peaks that we used. Okay, now it's finished. So we have to run the footprinting again for the cardiac cells. And when the comment is finished, this comment will generate the input, the output file for the footprints. And we will look at that file later once we finish this footprinting for the cardiac cells.
Li, in comparison, when you run this on the whole genome, how long does it take to run for one cell type?
I usually run this analysis. I mean, depends on how many CPUs you have. When I try to run these commands on the server, usually it takes 1 or 2 hours for the whole genome analysis. But I mean, the server has much more CPUs than my computer. So if you really want, I would not suggest to run the whole genome analysis on a local machine.
Or you do it overnight, huh?
Yeah, overnight, yeah, definitely.
So in principle, you can also run this with single end reads, or?
Sorry?
You can also run this with single end reads, right? Because there's this parameter paired end.
Yeah. We first, but comparing pair end sequencing, the single end will generate a loss. The prediction will not as accurate as pair end sequencing. So we also evaluated the single end and pair end in our paper, and we found that by using pair end, we can really improve the prediction. So we suggest to use pair end if you are able to generate the pair end sequencing data.
Is this because you have a good estimate of the fragment sizes that you get, and you said your model separates in fragment size?
Yeah, exactly, yeah. Because the fragment size also give you some information about the nucleosome positioning, and as we know that the factor will more likely to bind to the nucleosome free regions. So if we have the fragment size, we can use this information to improve the prediction. Okay, now it's finished. So next we can check what the footprints looks like. We can use head commands to only show the first 10 footprints, as you can see here. The first 3 columns define a genomic position, and this is chromosome name, this is start size and end size. And this column gives you the name for the prints. And more importantly, this column gives you the number of fragments around the predicted footprints. So we can use this number to rank the footprints and to select the most likely footprints. And next, we can generate the ATAC-seq signal per nucleotide. You can use these commands. So we first create a folder, and then we use RGT tracks. Oops. It's finished. So as I said, these commands will generate a ATAC-seq signal per nucleotide. So we can just import the signal. I will show you where you can find it. Load file. Go back to the thing. So go back to here. Go to the results. It's chromosome 21 tracks. And you can visualize the signal in your IGV. Since we only use the chromosome 21, so we need to go to there. Yeah, and we need to normalize a little bit. You can just zoom in to see how the signal looks like. And next step is motif matching. Since now we have generated the footprints, and based on these footprints, we want to figure out the transcription factor binding sites. So to do so, we needed to do motif matching. We first create a motif matching folder, and then we use RGT motif analysis to do the motif match. So copy here. And the database that we used for motif matching is Jasper 2020. And in this database, we have 746 motifs, which is quite large. Okay, now we have finished motif matching for stem cell. It's running for the cardiac cells. Since we have much more footprints in cardiac cells than in stem cells, so it will take a little bit longer. Okay, it's done. And now we have predicted the transcription factor binding sites for stem cell and cardiac cells. So next, we want to investigate which factors are either stem cell specific or cardiac cell specific. To do so, we can check the chromatin profile for each factor around for each factor between those two conditions. And to do so, we can use RGT hint differentiation commands to do the analysis. So again, first we create a folder for the results. And then we run the RGT hint differentiation commands. So here again, we are using HG38 as our reference genome, and this minus NC represents how many CPUs you want to use. So because my computer only have 4, so I will specify 4 CPUs. And here minus BC means we want to generate the bias corrected signals. You should be careful about these commands, and we only use it in this tutorial because when I test this command, most of the time, so when I try to run these commands, it takes a lot of time to generate the line plots. So we will try to not do that in this tutorial. But if you want to run, if you want to do the differential analysis, you should not specify this parameter. And the input for RGT hint differentiation is the motif matching results from those two conditions, as you can see here, and also the BAM file that the BAM file that we used to generate the ATAC-seq signal. You need also to specify the output name, and of course, and also the output location. So now we are trying to generate the signal for stem cells. When you start to run these commands, RGT will tell you how many CPUs you have in your computer and how many of them will be used. So on this computer, I only have 2, so I should not specify 4, but it's okay. So we will wait a little bit until this command is finished.
While we're waiting, Lee, can I ask you something? Now, we're doing this here with ATAC data because that's, the most common data in terms of open chromatin these days. But in your experience, does it work equally well with DNase-1-seq data?
I think, so we also compared ATAC-seq and DNase-seq, and initially we found that DNase-seq has a better performance. But now, because now the people have improved the ATAC-seq, which they called OMI ATAC, and we found that the OMI ATAC has the same prediction performance as DNase-seq. So now if you are doing, so now if you are using OMI ATAC, and they are the prediction should be similar to DNase-seq.
Okay, that's good to know. So if people, because I hear that sometimes, normally ATAC-seq is really easy to do. It's like a fast protocol, but then sometimes with some kinds of cells, people have struggled to get it working. And so if that was the case, and then you would have to do DNase-seq, then there shouldn't be a concern, right? I mean, if the protocol is nicely done.
And now we are generating signal for cardiac, and I hope it can be finished quickly. Okay. Since now we only use chromatin 21, so we also provide the completed results by using all chromosomes in your data. And you can, the results includes a text file. Okay, I will just finish. So we can go to next step. Okay, so the results includes a text file, which is differential statics text, and the results session 1 hint differential footprints. So you should be able to access this text file, and inside this file you will find the transcription factor activity. Next step is we can select some factors which are either specific to stem cell or specific to cardiac cell. So we can run this command to generate a PDF files, to generate a PDF file. So I will just show you how the file looks like. Go to results, session 1, hint, differential footprints. Click here. Okay, so as you can see here, we generate the bar plots to show you the factors which are significantly different between cardiac and the stem cells. And the y-axis is the TF activity. Sorry, the axis is the differences of the transcription factor activity, and the y-axis is the motif name that we used from Jasper dataset. So based on this plot, we can say, okay, for cardiac, there are many gatas which are specific to cardiac cells. And for the stem cell, we can select, for example, the POU5F1. We can also select ZEB1. And as I said, from this completed results, we also provide you the line plots where you can look at the chromatin profile around the predicted binding sites. So let's go to the line plots here. And we can just look at, for example, let me search a little bit, let me figure out GATA. Oops. Import the full name. Plot. So once you open the PDF file, you will see the line plots, which represents the chromatin accessibility around the predicted binding sites from stem cell and cardiac cells. So as we can see here, the chromatin is more accessible in cardiac cells, meaning that GATA3 is highly cardiac specific factor. And you can also check other factors which are stem cell specific. And finally, we will try to select those factors. So we can create another folder where we are going to select those cell-specific factors. Okay, one thing I forgot to mention is these R scripts will not only generate the PDF file, but also give you two text files, which include the factor name for each condition. So we are able to use it as input to select the cell-specific transcription factor binding sites. Okay. And now, since now you can try to input, you can try to visualize those binding sites by using IGV. So we load data. Differential motif matching. Let me see. PDF file. And then we can investigate how the factors regulate the expression of relevant genes. For example, we can check ATAC-seq. Let me zoom out a little bit. So, for example, we can find this factor T81 is only specific to cardiac cells, and might be regulate the expression of ATAC-seq. You can also.
Can I ask you?
Okay, go on.
Can I ask something? You know, I was wondering. I mean, this is an interesting example where you see that these motifs overlap peaks, and in the next part of the practical, we're also trying to answer how we can link those regions to genes. But now in this bar plot that you were showing, where this differential footprinting analysis is done, there were slight differences between some of the motifs. If you could go back to that.
Yeah, you mean here?
And yeah, this right here, and then we had this POW2 F3 and POW3 F4, and so now, in your opinion, is it? I mean, this ranking here, for example, POW3 F1 has the strongest negative, but the strongest change in the direction of HECs. But in your opinion, is this something one should interpret, or is it just saying that, well, look, all of these factors here are in principle, more in open sites?
Yeah, sure. Because the motif that we used the motif of these two factors are very similar. So by only using the DNA sequence, we are not able to distinguish between them. Meaning that this is not, those two factors might be like so one of them might be false positive, but that is not distinguishable for us. So if you really want to link those factors to genes, we also need to check the gene expression data to really confirm that one of them is specific to stem cells. So yeah, so does this answer your question?
Yeah, absolutely. I'm just thinking that now, because you have a way of, because you compute this for all of the hundreds of TFs, right? For which you have motifs. But now in this plot here, you only show the ones where there is a significantly different activity, right? You're doing a test to say, well, how different are these scores for each of the TFs?
Yeah.
And so in your experience, or I don't know if you've compared or so, but the significance test, what is this, a Wilcox rank sum test, or what was it?
It's a Z-score test.
A Z-score test. Is it, basically factors for which this test is not significant? Do you still see that, if you now look at ChIP-seq data or something, that these factors are important? I'm basically asking how, how sensitive is this test?
Yeah, sure. So this test, in my experience, this test will only select like top 5% of the motifs that you test. So it should be possible that you generate some false positive or some false negative. And regarding the sensitivity of this test, we haven't done some experiments to evaluate the sensitivity. And we also use the number of binding sites to select the factors. So hopefully we can decrease the number of false positives. Yeah, so I think now we have finished the practical one. If there's no questions, then we will go to next part.
Sure. If you unshare your screen, I can share mine.
Okay, you can just directly share your screen, and let me stop the share. Okay, I think now you are able to share your screen.
You can see the slides, huh?
Yeah.
All right, good. Well, then, I hope everyone was able to follow so far. Please let us know if there's any problems. Now we're going to, in the second part, we're going to dig into the questions. What are the questions? How, which transcription factors are now involved in regulating genes of interest? So individual genes, as well as sets of genes that are differentially expressed, And you know, one essential question that we're dealing with is this. It's really the question behind, well, if there are enhancers or regulatory elements, to which genes do they belong? You know, and it's not an easy question to answer. I can tell you that. But we're going to talk about the different options you have. And one aspect of this is then what you can do then once you've assigned possible target genes, you can ask like, what are the different biological functions of those genes? And by this, you can maybe reason what is the role of the transcription factor in, or what are the roles of the transcription factor in your system that you're studying. So let's talk about the question how we can predict target genes of regulatory elements. So, you know, enhancers can be really far away from genes, from gene promoters, because there's huge mediated complex, which is like a bunch of factors, and it helps to like stabilize, basically forming a bridge from enhancers, you know, to promoters. And you know, probably know that, you know, enhancer can also regulate many genes. And so this can really get quite complex. I mean, there's genes that have, you know, tens of enhancers, and to know where they are and when they're active is really, I think, an active field of research. And so I think by the virtue of generating these data that you know, epigenomics data, you have a chance of finding those enhancers, specifically also cell type specific enhancers, which may be really important. And so, you know, how now do methods use this kind of information, also like footprints or so, and integrated gene expression? There's really many different approaches that people have presented, and I can't really go over all of those, but here's like some you can like, you know, basically look into, where they basically use chromatin data and also TF information in an informed way. And particularly now, if you want to link this to gene expression, there's a couple of papers that you can also have a look into. And so today I'm going to talk about our approach. Is called T-EPIC, which is a software that can do many things around the integration of epigenomics data with gene expression. And so we will talk about different approaches for linking elements to genes. And so I think the two most naive, or better to say the most common, are window-based and USGene. And then the third one would be using chromatin confirmation data like Hi-C data or ChiaPet or something, or association-based. Okay, now in the context of this T-EPIC software that we're using, I want to discuss one conceptual difference on how you can use PWMs for predicting motif occurrence. So what Lee has been telling you now, and what is also used in HINT, is this idea that you have a PWM, and then you go with this PWM along a sequence, and then you ask, you know, which of the sequences are basically significant under some statistical model. So, you know, there's different ways how you can do this, and, you know, you can read up on how HINT does it. But basically, it's like, you know, based on the information content on the motif, and then on some assumption on the background motif set, you want to say whether a certain sequence is likely to occur by chance. And then sequences that are a really good fit to that motif, you know, basically the consensus here, like what you see, these are your motif occurrences that the model would predict. So these are the hits, basically, that you also find as output in the HINT files. You know, I mean, that's a most widely used idea of how you use these PWMs for motif prediction, and that's really fine. But there is an alternative way how you can interpret those models. If you think about it more from a biophysical aspect. And so here the idea is that, if you think about it, that transcription factors, you know, they're not making binary decisions in the sense that they bind the DNA and then they stay there indefinitely. You know, of course, in the cell, it's like an on-off situation, and you can express this with sort of like, you know, basically computing constants of like, you know, how often the transcription factor will be bound to the sequence and how often they will be apart. And there's methods for doing this. One of those is called TRAP, which essentially takes a PWM and then converts this into an energy matrix. And then what it does is that you basically, instead of looking for one individual hit or several hits along the sequence, it will sum up the contribution of each individual base pair, basically moving the PWM along the sequence. And this is what they call an affinity. So basically for a sequence, you will get an affinity, and it should resemble, or it should estimate, the average number of bound TF molecules in that region. Yeah? So it's basically, you can think of it as a biophysical inspired statistic estimate. And so the higher that affinity, it would mean that more positions along that sequence are likely to high occupancy sites of the factor. And so now the good thing is, now instead of hit-based, where you have integer numbers of 0, 1, 2, 3, whatever in a sequence, you have like more numerical values. And we found that this is very, first of all, it captures more information, because also low affinity sites, which are basically, you know, just below the threshold of your statistics you use for significance calling in a hit-based approach, you would still you know, contribute. Okay, and so now how does this work in T-EPIC? So the Windows-based approach is basically like this. You start with the gene annotation. So here's the start site of a gene. Now you do, you know, peak calling or footprint calling, like we just heard about from Li. And then in these regions, you use each individual transcription factor, PWM, and then you compute those affinities using the trap software that I just mentioned. Okay, so basically these are numerical values. And these values are not directly comparable between different TFs, but for one TF, you can compare them. And I should also say it's also influenced by the length of the region, yeah, because you're summing these contributions. So, so if you do this, then basically there's a way of now subsuming this information for each individual gene. And this is explained in this formula here. So what we want to have is for each gene, we want to have an estimate on how likely that gene is being regulated in a certain cell type. And so that's basically the affinity A for a gene and for a transcription factor. And so what we're doing, we're summing over all of those regions, so the peaks or footprints that you want to link to the gene, okay? And then the essential part is this affinity score that I just explained. That's what you get out of TRAP for each TF in each region, yeah? So that's basically here 3.4. Then there's also, I mean, T-EPIC has the you know, possibility that you're using additional information. So for example, you could include the magnitude of the signal, of the ATAC-seq signal, because that's also, you know, I mean, if you have like a larger peak, it's more likely that in many cells of your sample, the chromatin is open compared to if you have a smaller peak, especially after the, you know, bias correction that Li explained. And so you can also include this as a parameter in T-EPIC, but you don't have to. And then now, of course, one important problem in this is that how far do you want to go, right? And I said, this is a Windows-based approach. And so what we're doing here is that we're saying, well, you know, the more further away you are from the transcription start site, the less likely is it that this region is actually having an impact on the gene. Why is that so? Well, because people have observed by looking at high-C data that you know, the likelihood of getting in contact with another position along a chromosome, you know, decreases basically exponentially with distance. So if it's really far away, then it's less likely that these two DNA pieces interact. Yeah? Of course, it can happen. There are always exceptions, but on average, it's very unlikely. And so essentially, this part of the formula here downplays regions that are further away. And so this was originally proposed by Yang and others here for ChIP-seq data, and we had adapted this, you know, in the use for open chromatin data. Okay, now and then basically in this way you aggregating in a window size that you define all of those peak or footprint regions, and we get a score for each TF. Okay, and then now you can use this to now link them to gene expression. So one way that we always like to do is to use machine learning and in a regression setup, for example. And so here you would have a matrix where if you just have one sample, which is the thing I'm just shortly want to talk to you about to tell you one aspect, is we basically would go over all genes. So we have n genes and we have n manufacturers. Now each entry is a score that I just explained, one of those AGI values. And here is the gene expression. So for example, you measure with the microarray or RNA sequencing. And so what we're trying to do, we're trying to solve this regression for this beta vector here, minimizing the error of these affinities times the to be estimated beta to directly basically explain gene expression. Okay? We're using here elastic net regularized regression because to basically adjust for the fact that we could have many TFs, more TFs even than genes that we're using. And yeah, and so in this way, then you can look at those beta coefficients. And basically the reason why I'm mentioning this to you, because I want to give you a bit of an idea what's the difference of peaks and footprints in terms of understanding gene expression. And so here, wait, I should basically say this. So, you know, peak calling was done with Max2, as you know, and now you've hopefully were able to use HINT to call those footprints, which are always smaller regions compared to the peaks. And so what we've done here, we've used 4 different ENCODE cell lines, which are depicted here, these different symbols. And we've compared this kind of, the whole computation, so the affinity gene scores for each gene, and then we've run the linear regression to get an idea how good we can estimate gene expression over like all genes. So what you see here is the correlation of that regression model on test data that was not used. So the higher the better, so the best would be 1. That's a bit unlikely given that, you know, this is actual biological data. But what we're comparing now is to use peaks in the vicinity of genes or footprints from a HINT-BC in the vicinity of genes. And so there's always 2 different setups. We have actually, we have different setups here, different window sizes, so a 50 KB window around the start site or a 3 KB window. And so these are the colors. And so what you now can see here is that overall, peaks versus footprints is very similar. Yeah? So there's some slight differences. For example, we noticed that the HapG2 cell line, you see that peaks worked actually much better than footprints for some reason. Yeah? But for the other cell lines, it's actually not exactly the case. For example, for GM cell lines, it was the other way around, that footprints were a little better than peaks. Okay? Whereas for the H1, HESC, it was actually, it didn't really matter much. And I think that's remarkable because footprints are much smaller than peaks. And so it means, or at least it suggests at the very least, that when we're doing this footprint analysis, we're really capturing the essential regulatory part often of those peak regions. Yeah? And otherwise, it's, you know, less likely that we'll get to similar results. But it also means that in terms of like expression learning, at least, I can't say always use footprints, always use peaks, but definitely using footprints makes sense. Okay, so that was essentially this window-based approach. And so, you know, basically starting from the start set of genes, you make a window around those peaks and then you just associate them to that gene. Now, another very common and easy approach is the so-called nearest gene, where you say, well, I'm basically looking in the vicinity of a peak or footprint and then assigning it to the nearest gene. Yeah? And so the difference is, the fundamental difference is that here, this is a one-to-one assignment. So each peak or footprint is assigned to only 1 gene. Whereas in a window, if there's, you know, a peak could be assigned to many genes, everything in the vicinity of that window. But the advantage of nearest gene could be that peaks that are really far away, I mean, much further away maybe than the window we looked at here, can still be assigned to the correct gene. So I wanted to use now the Portal app here, and because I posed a question asking which target gene identification method is better in your point of view. And you could click either on window-based, nearest gene, or both have problems. So I'd like you to, you know, click this now. And then in like a minute or so, we're gonna see what, you know, people think. Okay, so 9 people voted. If you can, please, other people also vote. Okay, so I'm gonna stop the vote now. So we had 10 votes, and 80% of the people said, well, both have problems, and 10% said either window-based or nearest gene. So, well, let's have a look. Let's compare those two in this gene expression prediction task that I just explained. So here's a, here's an example scatter plot where each plot is a gene now. And we've learned this gene expression model either using the window-based, the 50 kb window around start sites, or the nearest gene approach. This is in HeLa cells, yeah? DNA is one seq actually. So now what you can see is that like overall, both methods agree, right? We're using the same DNA data, but there's really quite some difference on individual genes, yeah? So actually, there isn't a very clear winner. On average, we found on most of the data sets we looked at that window-based is doing a little better than nearest gene. But as you can clearly see here, it's not like a clear trend. And so now why is that, right? What could be the problem? So for example, here, if you look at the HINT-1 gene, where the nearest gene model works much better than the window-based model. So this is here now, I'm showing IGV plot of the HINT-1, and you see the locus. This is a really large window, 109 kb, yeah? And what you see, there's one other gene, but the rest is pretty much not annotated, okay? So I'm not showing you the DNase data, but what we've noticed is that genes that are very isolated and sort of like very, like gene deserts, so to speak, the nearest gene model works pretty well often because it's a pretty good bet to say that, well, this, even far away peak is, regulating that gene because there's simply no other gene. Okay. Now, on the contrary, for example, the APO-A2 gene here, which is doing much better in the Windows-based model, that is a gene that is in a very gene-rich region of the chromosome. So this is now an 18 kb window. So here's the gene, okay? And you see there's like, just in that window, there's like 4 other genes, okay? Within. So if you're doing a 50 kb window, as we do in this experiment, actually we include all of those genes that you see here and more maybe. And so you see how both of these kinds of approaches are really imperfect because they're not tailored to, to basically the situation of the genome, like, and other maybe chromatin differences. But anyway, what I want to say, if you have to choose one of them, I would say on average window-based works best. But if you're interested in particular genes, always look at the genes, see what's the vicinity, where are genes and how peaks are changing and so on. Now, of course, if you're lucky or you work on a cell type where people have actually measured DNA contacts with Hi-C data. So I just want to briefly mention this. This is a protocol where you're linking regions of DNA that get in contact. So this is basically you're crossing this and then you cut it and then you sequence those parts. And now you have the 2-part sequence that basically get in contact. And then you can generate from these contact matrices, and you can look in larger regions of the chromosome, whether it's likely or not to have contact. So everything, when you see a red dot here, then these 2 different parts of chromosome 14 here, for example, are getting in contact with one another. And so if you have this data, so if there's like a related cell line, you can actually use a very recent approach, which is called the ABC score, which essentially says, well, if I have activity of, for example, H3K27 acetylation, which is a histone mark, or you could also use ATAC-seq or DNase-seq. You can take that activity and you can multiply it with the normalized number of contacts that you get in this Hi-C data. And so in this way, you can make an estimate whether a certain region would get in contact with that gene, and by that logic, maybe regulate that gene. Yeah, so if basically you have data or you're producing this kind of data, then I would suggest to look into this paper. It's a very simple approach. Okay, but you may not often have Hi-C data, and so one other thing that you can use is basically association-based maps. And so I'm going to show you here, or explaining basically the idea of this. So when you've gathered a lot of open chromatin data, which is shown here, so this would be very different cell types or a lot of different inductions, and you've measured open chromatin like DNase-seq or ATAC-seq, and at the same time you measured in the exactly same set of cells, you measured gene expression, like gene activity. Yeah? Then what you can do, you can use machine learning to basically see whether there is an association of a certain region, which is like open, like you see here, and the gene expression. Yeah? For example, here in these first parts, there's more activity of those genes. And we see here, there's a region where there's always an open chromatin region here and here. There's generally much more activity. around the promoter. And so you can basically design this as a machine learning task also. And people have done this in different ways. We have made a software that's called Stitchit, which can essentially do this. So you give just a set of paired data, and then you can learn the location of regulatory elements and to which genes they link. Yeah, and we have done this using the blueprinted roadmap data sets, which contain cell types and tissues, always RNA-seq and DNA-seq of the same samples. And then what we've done, we've used the Stitchit method that learns these associations. And we ran it in a way that for each gene, we would use 100 kb upstream and 100 kb downstream of the complete gene, and then also including the gene body. So basically in all of that region, we said, oh, are there regulatory elements that are associated to gene expression changes? And this is something that we put into a database, which is called epiregio, so epiregio-DB. And epiregio contains 2.4 million regulatory elements that we've inferred in this way from the data that I mentioned here, which are linked to also putative target genes in human. Okay, and so conceptually, it's like you have a gene G, and now you have in these really 200 kb upstream and downstream areas, you have regulatory elements. And in the database, we store information what's on the activity of those elements and the location, of course. And now you can, for example, go to the database and look them up, where they are and what their activity is. And so I'm going to show you shortly, at least that's what I'm trying to show you the database. So this is the database, it's just epiregio-DE, okay? And so now you can see, for example, you can go here, you can look for a gene that you're interested in, no idea, TFGN, type in the name, get the gene ID, and then you query the database. And then you can, now these are all the regulatory elements, you just get a list, but you could also select here, for example, a cell type that you're interested in, and then you could see the activity of those. So you can do, many different things, and I, encourage you to have a look at it. I think in general, the question that you can address is, what are the putative regulatory elements for a set of genes? So you can come with the genes and then you can be given the regulatory elements, or you can come with the putative regulatory elements, or or footprints or peaks, and you can ask, well, do footprints overlap with elements in the database? And then to which genes do those elements be, are associated? And so basically you can, just with peaks and footprints, you can generate a list of target genes in a sort of I would say, informed way in the context of the database. You can do this with a web server, I just showed you, or you can do this with a REST API using, for example, Python's request module. And this is also something you will do in the tutorial. Okay, so now, let's ask how we can use gene expression and TF data to understand differences now between cell type. Okay, so so basically, that's a very common question, right? When people do epigenomics data like ATAC-seq, you often want to see how which transcription factors are now binding in those footprints, for example, and then regulate gene expression differences. And so, so let's say you have a set of differentially expressed genes that you obtain from analysis of RNA-seq data. Then one thing that you can easily do with different software that's available in the web is you use so-called TF enrichment analysis. So what these methods do, they basically annotate the promoters of genes and ask, well, are there binding sites in, is there basically enrichment of binding sites of promoters in upregulated genes, for example, compared to downregulated genes? Like you see here, the red TF is only upregulated genes. Yeah? And that can be helpful, but we know that there's lots of enhancers that are not at the promoter, and these may be relevant in the system where you're looking at. And so as part of this T-EPIC software, we also have a workflow that's called Dynamite, which basically essentially does this. So if you have footprints and here, then you can annotate those footprints with binding site with this trap method, and then you can aggregate the data on gene level, as I explained in the first part, to basically get cell type specific TF gene scores. Okay. And yeah, and then you can use this in a differential analysis. So now what's the difference to the thing that Li just explained, where we're going to do this, where we're doing this differential footprinting analysis? Now, if you remember you know, what Li has explained, if we have two types of samples, like here, and so let's say these are footprints called here, yeah, two, these are the footprint locations, and there's also binding sites of a TF that we're interested in, right? Then in this previous analysis, you would just take all of those binding sites that are in footprints, and now you would compare the basically protection score between the two samples, right? So here, for example, is higher, more DNAs were attacked than here. But you wouldn't know, you know, whether these are, you know, at the locus of genes. So it's basically just genome-wide. And so it's an interesting analysis for seeing, well, which factors seem to be strongly changing. But of course, you don't have a direct connection to gene expression regulation. And so now here in this dynamite analysis, we're making that connection. And so the way that it works is you obtain ATAC-seq data or DNase data or so, or you can also use histone chip-seq data for your two setups, for example, control and disease, okay? This is then, for each of those setups, you're going to create, you basically run T-epic, okay? And that's going to give you TF gene scores. Yeah, we don't want that now. And from those, we're going to make then kind of differential score I'm going to show you. And then with these differential scores, we're learning a classifier that should be, that we're training for differentiating the up and down regulated genes that you've obtained with, for example, RNA-seq data. Okay, and so basically through this pipeline, we're linking changes in binding of transcription factors in the vicinity of genes to changes in gene expression. Okay, we call this dynamite. And so, like, you know, one common way how I think it makes sense to do this, because, you know, basically what kind of ATAC-seq information you're putting into this T-epic software here is. One common thing that you can do is you can, you know, you first call footprints, and then you look at differential p-calls. And so, for example, here's a region where you see there's much more in this sample than in this sample. And then you're using those regions as input for the T-epic analysis, because these are likely the ones where there's really a change, which could explain also differences in gene expression. Okay, and you can play around with this and just see what works better. And so, what's basically the underlying setup in dynamite? So if you have the 4 changes of differentially expressed genes, these are going to make up like 2 classes, up and down regulated based on the 4 change. And then this part here is now this TF gene derived matrix that dynamite computes, which for each TF is going to compute a ratio of the affinities for the 2 you know, sets of samples you have. And so what the classifier is trying to do is say, like, well, you know, does any of those columns have information on, you know, the gene expression status of the gene which is in the row? And so if you see basically an association that, let's say, TF2 is always really high when it's like an upregulated gene, or TF1 is really low when it's a downregulated genes, any of those, then the classifier will pick this up and report it to us. Okay, so so how do we get to this matrix? I said we run T-epic on each replicate data set. So this is basically one output of gene scores for one replicate. So we get these 2 replicates. We create a mean matrix. So basically, you know, take for each corresponding entry, we do compute the mean. And then we're going to have this for the 2 types of samples, for example. And so we're then simply doing a ratio between those values to basically see, because we want to explain differences in expression. And so we want to see that the binding affinities change, right, for that gene for a certain factor. Then we think that new binding sites have opened up or have been closed in the ATAC-seq data that we used. So I'm going to show you one example of this using data set of CD4 plus memory T cells, where people were interested to understand the differentiation of naive T cells, because it wasn't really clear whether from this naive T cells you would make the different types of cells, like central memory T cells or effector memory T cells, or whether there would be a linear progressive model. Okay? So the context of this project, we used epigenomics data to basically do this kind of prediction. And so we did 3 different types of comparison. Here, these are 2 different types of T cells. And now what you see here on the y-axis is the mean test accuracy of the classifier that Dynamite uses, which is a logistic regression classifier. And so if accuracy 100% would mean that we can always perfectly say what's up, what's down regulated. So this is not what we're achieving, but we're achieving, you know, decent result on that data set. We also did this on another mouse data set at the time, and there it was better. So it depends on your data set how good you'll be able to separate basically differentially expressed genes. Also, it depends obviously on the method to call differentially expressed genes and so on. But it gives you already an idea how informative are the TF features that you're computing from the epigenomics data. And then you can, as output of that software, you get the feature coefficients of that logistic regression classifier, and you can rank that. And this is what we've seen here. And so it basically proposes, a number of transcription factors that are related to, for example, the differences between naive and memory cells. And we actually validated this experimentally. And we could, one of those factors here, FOXP1, this was done in this paper. And what they've shown, if you knock out FOXP1, then these cells lose their naive status. So now one thing I want to mention in all of these analyses that we've done so far, it's useful to remove non-expressed TFs. I mean, Li already said it. And so what you're also going to do at the start, going to remove TFs that don't show a high expression or like any expression really in any of the samples you have. That's just a way of filtering the things that are not so interesting. Good. So with this, I'd like to thank Nina, Shiva, and Dennis for preparing this part of the tutorial, and also our funding agencies for the funding here. And so now we're going to go through these two practicals where you're going to use Dynamite and then link footprints to genes. Are there any questions? Current questions, none. So I don't know if you have one. Quickly wait, otherwise switch over now. Feel free to post the question if something comes to mind or so while we're going through this.
Yeah, okay, if there are no questions, we can start with hand on 2. Just share my screen. We can now just a second. Okay. You can now see my screen, right?
Yep.
Okay, this one. You've seen the GitHub page, right?
Yes.
Yep.
Yeah, okay. Okay, yeah, for practical 2, as Marcel said, we're now going to use Dynamite to infer transcription factor that might be related to the gene expression changes between the 2 cell types we're actually interested in, which is in our case these human embryonic stem cells and the cardiac mesoderms. And yeah, first, it's important that you ensure that you're in the correct folder directory, which is actually here. And the data epigenomic analysis tutorial 2020. And yeah, in our first step actually, we want to get rid of all these transcription factors which are actually not expressed in the human embryonic stem or the cardiac mesoderms. And yeah, therefore, yeah, we're going to use a script which is called extract PSEMs of expressed transcription factors. And yeah, this will do the job. It actually takes as inputs the gene expression values from the human embryonic stem cells and the gene expression values from the cardiomyocytes and the mesoderms. And yeah, we actually pre-computed that data for you. We took the data from this paper Marcel was talking at the beginning, and then we run Salmon to do the quantification. Yeah, and then we also need a list of all the available transcription factor binding motifs. Yeah, we also need a mapping of the sample IDs to the gene names, and yeah. So now to run the script, you can just copy the command here, and yeah, then run it in the copy it to the terminal and just run it like this. And then before doing so, you also have to create a result file, the result folder in the result folder using this command. Yeah, I already did that, so it worked for me, but I guess you have to do that. Yeah, and then yeah, we created the output file where we now stored all the transcription factor binding motifs with which are actually active in either the embryonic stem cells or the cardiac mesoderms. And this file you can now find in the folder results season 2. Yeah. Yeah, that's actually all I want to say here. And in the second step, we want to intersect the footprints from HINT with these differentially with this differential ATAC peaks. Also, as Marcel explained, and yeah, the idea is here that we provide dynamite only the regions where we expect the transcription factor binding TFs to bind, and where we also see a change in the opening chromatin profiles between the two cell types. And yeah, to do so, you first have to pre-process the data in such a way that the footprints are in a proper BET file format, because we want to use the BET command, BET-tools command intersect to do the intersection. So yeah, therefore you have to perform to run these two commands here. This one. You can just do that. Once for this human embryonic stem cells, and then also for the cardiac mesoderm. Yeah, this should work. And then the result is always stored in this results season 2 folder. Yeah. So, okay, and after doing so, we then can use BET-tools to do the intersections. And it's just a basic intersection command, nothing special. And you also have to do that for the human embryonic stem cells and the cardiomyocyte cells. Okay, and yeah, one thing about these footprints, yeah, they are rather short. And to probably compute these TF binding affinity via T-ATAC, we need to extend these regions, but they are at least 20 base pairs long. So in this next step, we want to add 10 base pairs before and at the start and at the end of the footprint, so that these regions for sure are longer than 21 base pairs. Yeah, actually this is really important, otherwise T-ATAC is not working properly. So keep that in mind. So you can just copy these two commands, maybe one. Okay. And with that, we are basically now ready to run Dynamite. And yeah, as Marcel, he basically said everything about Dynamite. And this is a two-step approach where you first calculate these TF binding affinities, and then aggregate those binding affinities to these gene scores using T-ATAC. And in the second step, we then use this logistic regression classifier to learn the changes in these TF gene scores between the two cell types. And yeah, we actually provide you a script which will do these two steps. And yeah, before we can do so, you can maybe create this result file folder for Dynamite with this command here under step 2. I mean, I already did that. And then we changed the directory to the EPIC folder. Yeah. And here you can now see the, I mean, I already prepared it for you. It's basically the setting file we need to call Dynamite. And yeah, here you can see that Dynamite uses as input these footprints which we prepared, the extended footprints which we prepared. And then here is differential gene expression data. Yeah, so yeah, we use these ATAC-seq data from the paper which Marcel was talking about before. And yeah, I determined the differential expressed genes using DAC2. And I mean, you need this file as input because they are the regions. Yeah, it's just the differentially expressed genes basically. And yeah, then you need to provide an output file. Here you have the option, if you already have pre-computed results of the EPIC, you can also provide the path here, and then you can skip the call of the EPIC. But we don't have these files, so we have to run the EPIC. And then, yeah, you can also specify a set of parameters for the EPIC here. For instance, where the path, where you can find this codes, or where you have to specify the path where you can find the genome version we're going to use. Then you have to specify which transcription factor binding motifs we're going to use. And here we also use this file we just created, where just list the binding motifs with the active transcription factors or the expressed transcription factors. Yeah, and then for the logistic regression step, we're going to use this cross validation with 6 outer and 3 inner loops. Yeah, and an alpha step size of 0.1. And yeah, to run the script now, you can just take bash run Dynamite and then provide the configuration script, and then it will run. Okay, are there any questions so far?
Nothing posted.
That's good. Okay, until this is running, I thought we can already go over the result because I pre-computed for you, so I thought, I mean, we don't have to wait for it. Yeah, okay. Okay, the result is stored in, and this is the shared Docker container, and then you can go to the epigenomic analysis tutorial, and under results, season 2, you will find this Dynamite folder where, yeah, Dynamite, all the results of Dynamite are stored. And then you will see here 3 directories. I mean, this affinity, affinity is just, there's basically the result of the EPIC that is stored there. So yeah, you can have a look at it, but yeah, basically it's just the output of the EPIC. And in this integrated data set folder, there you will find the basically the feature matrix and the response, which is basically used as input for Dynamite. And the most interesting here is this learning result, so you can see the output of Dynamite. And yeah, yeah, in general, I mean, there are several result files, and I just want to go quickly over them. Yeah, the first one I want to talk about is this performance result file. It produces actually a bar plot for it, but it also has the result as a plain text file, this one here. And if you open it, yeah, you can basically see the accuracy on the test and trainings data set, as well as on the F1 measure. And yeah, then for each of the outer loops, it performs a confusion matrix. I can maybe also open that one. Yeah, this is basically just a confusion matrix, as the name is saying, where you show the model performance by reporting the true positive, false positive, as in this true positive value, the false positive, the true negative, and the false negative value. And yeah, this is, you have these files for each loop actually. Yeah, and then basically the most interesting file is this with the regression coefficient output file, where actually for all transcription factors with the non-zero regression coefficients are actually shown here. And yeah, this is basically also the main result of the dynamite. It shows you basically which predicts the involved, yeah, shows you basically transcription factor where we predict that they are involved in explaining the changes between these two cell types we're actually analyzing. And yeah, all of this also explained in our documentation. If this was now too fast, you can maybe have a look there. Yeah, for me also the dynamite one is now done. I hope it all worked for all of you. Not sure if anything else to say. Yeah. Okay.
Can you open the file again with the result, the accuracy, please?
Which accuracy?
Yeah, it's still open there.
This one?
The performance bar plot.
Ah, this is open, yeah.
Yeah. So, right, so here we give the accuracy on the training set, which is the black one, and then an orange on the test accuracy. That's the one you should really look at. But we see that the training accuracy is like actually significantly higher than the test accuracy. Yeah? So it seems like it's like a slight problem of overfitting, I would say. I mean, overall, the accuracy is not incredibly good. I mean, Lee had also mentioned that the HINT data, right, is not doesn't have so good quality, so it could be due to this. You know, don't really know. So we've definitely seen better results. But if you go now to the regression coefficient, what I noticed is that you know, GATA3, GATA6, these were also among two of the factors that Lee had talked about, where we saw really large differences or strong differential binding according to footprints genome-wide. So actually we can, now from this analysis, reason that they are in fact really are important for regulating at least a set of genes that were, you know, put into the dynamite analysis. And there's also another feature here on the left, is called peak counts. So T-EPIC actually, I didn't really say that, but it has also, it can not only compute these TF scores, it can also just look at, well, how many peaks are there or footprints are there in the vicinity. So actually this is not peaks, this is footprints because we gave it footprints, but it's always called peak counts in the output. And so you can also just use it to count basically for one sample, how many peaks are there in the vicinity of a gene. Yeah? And what we'd found is in previous studies that there's a lot of information. Just knowing the number of peaks, that's basically what you're seeing here, is a good is an informative feature for the classifier, right? If I have suddenly 5 peaks and before I had only 1, then it's likely that the gene is more expressed because most of these peaks have a positive association with expression. And so, yeah, so this is what the classifier is telling us, that there is a lot of changes just due to the peaks. And yeah, and then you can see also ZEB1 was also something that came up with the differential footprinting analysis. And there's, you know, a couple of other factors that you get now. And, you know, and so I think this kind of analysis strengthens the, I would say, interest as, you know, to reason in more detail on some of those factors like COX-B4 or so, and trying to really understand which genes are, you know, what are the genes that are regulating and how is that connected to the processes that we're investigating. Yeah. So are there any questions for that part? Well, if not, then I think we should move on to the next part, the practical. Dennis will take over. Who has the most appropriate background of all of us.
Okay, so now you can hear me, right?
Yeah.
All right. Okay, so I will conclude now with the third hands-on part, which is the identification of target genes of specific regions. And we want to look at specific TFs, where we would think that these are now responsible for driving this differentiation from the embryonic stem cells to the cardiac mesoderm cells. So this is the box here. And what we will use as regions are the footprints, or more specifically, the motif hits of a transcription factor, which we can choose. Yeah, exactly. Then we will use the three different approaches shown here, look at the target genes that we get as output, and then compare them with the genes where we know that they are differentially expressed because we already did a DEC2 analysis with the RNA sequencing data for this data set. So I will also go through this with you. First of all, make sure that you're on the right path. So in this data folder and also the epigenomic analysis tutorial folder. And as next step, we first need a results folder, which I already did. So if you didn't do it already, please create this folder. And the next step is that we create a file where we have our regions in. And as I said, we will choose one specific transcription factor. And just as an example for today, we will use ATAC-6 because it already came up in both analyses. So we'll go with that one. If you want to have a look at a different transcription factor, just replace every ATAC-6 in this documentation with whatever transcription factor you want to look at. And from the HINT analysis, we already got the locations where we think, or where it is predicted that ATAC-6 is binding. And these motif hits are all in this file here. And we will filter this file for the hits that are recorded as a ATAC-6 hit, so ATAC-6 bound there. And then we will intersect these motif matches with the differential ATAC peaks. So the idea behind this is to get the positions where ATAC-6 is binding, but also in regions where we think where there is a difference between the two cell type conditions which we are comparing. So this is what this script does. Just copy paste it. And then you have your output file. So initially we had for almost 5,000 motif hits, and approximately 1,000 of those also intersected with a differential P. So these 1,013 motif hits are now the ones where we want to predict the target genes for. And for this we have three different approaches, as I said. First of all, we will start with the nearest gene approach, which uses BET tools, or more specifically the closest commands. So for every region, we will look at the closest transcription start site and receive this as target gene. So again, copy paste, and you should have now a text file with the target genes. And I can also show this. So I already did all this. For you, the files will appear one by one. So we just created this file here, nearest gene, which is a list of gene IDs. So these are the ones that are the closest to the ATAC-6 motif hits. As next step, we will use the window-based approach. So instead of using the closest gene, we will take a 50 kb window. You can also choose a different one if you like. We will now go for 50,000 base pairs around the TSS. So execute this. And you will also find a text file with the target genes in the window. And the third approach is the association-based approach. And for this, we will use the data behind epiregio, about which Marcel already talked. So it's not location-based, but association-based. And for today, we will not use the website. You can also use the website, try it out if you want to. But we will use a Python package called requests, because this can pose HTTP queries. So our epiregio website has a REST API. And what you can do with this is that you can create URLs, send it to this REST API, and it will return to you the data that you requested, which is pretty useful if you want to include it into your script, so you don't have to exit the script, go to the website, and then process it further. Yeah. And for these 1000 regions, this will take 1 or 2 minutes approximately. Are there any questions until this point?
Nothing on the portal.
Let's take this as a good sign. But as I said, I already performed all this, so we can already look at the results because this last command here will create some more files, which we will go over now. First, we get 2 plots. Yeah, because what we want to do now is to somehow get an idea of how good, so to say, our target genes are. We will look at how many differentially expressed genes are among our target genes. And for this, we will get an upset plot, which would look like this. I mean, this is more or less like a Venn diagram, but with a different way to depict things. And it is quite handy if you want to understand where to look. So what we have here is that we compare 4 different lists of gene IDs. We have our 3 different approaches to find target genes: nearest gene, window-based, and association-based. And also we or differentially expressed genes with a cutoff of 1%. And we had 7,450 differentially expressed genes. And you see here the numbers of target genes which we identified with the different approaches. And what we already see is that the one with epiregio has the lowest gene ID count. Yeah, which is also caused, I mean, by the way it is determined the target genes. Yeah, and the most interesting intersections are the ones that are shared with the lowest column here with the differentially expressed genes. For example, if you look here, I hope you see my pointer clearly, is the intersection of the differentially expressed genes and the nearest gene approach. So what we see is that 57 genes of those nearest genes are also in the differentially expressed ones. So this is what we're hoping to see, that we also find target genes that are also differentially expressed, because this could explain why this specific transcription factor always was shown in our analysis before. Then we can also have a look at how large the overlap is with window and association based, or also which of them are shared. So for example, 11 are found in every set of genes. Yeah. And you will also find a bar plot inside of this folder. Where did I hide it? There it is. Because if you look at this and in this upset plot, you see that the number already varies. To get a different view on it, this bar plot here shows the fraction, the percentage of target genes that are also differentially expressed. Yeah, and what we see here is that the association-based approach has the highest percentage of DE genes. So more than 20% of all the target genes that we found were also differentially expressed, which kind of is a good thing. Yeah.
So this kind of suggests that when you use the association-based approaches, and in this case, the epiregio database, I mean, that you have a better specificity, right? But you may be losing some of those target genes. So exactly. If I would always start with those and say like, well, okay, there I have a bit more evidence, so I'm maybe a bit more sure. And then I would ask myself, okay, how should I now take maybe you know, should I take all of those approaches and use gene window based, or just, you know, window based? And this is then up for decision by the user, essentially.
You can also, what we create in the output folder is further text files. Because you might be interested to see which genes are those that intersect here. So we'll find 4 new files, which is, for example, association genes, the E genes intersection. And in this file, you will find all the genes, all the target genes from the association approach that are also DE genes, so differentially expressed genes. That would mean the 37 here, in addition to the 4 here, these ones, and the 11 ones here. And on top of that, there's also a 4th file, which is called approaches merged. So as Marcel said, it's difficult to decide which method is really the best. Maybe you want to have a broader range of target genes, or you want to be more specific or hope to be more specific. And this is why this file now contains the pooled target genes of all 3 approaches intersected with the differentially expressed genes. And with this, you can do further analysis. For example, you could go to a functional enrichment analysis tool, post this there, and see what is the output. And this is exactly what we will do now. For example, G profiler is pretty handy. So we can just copy the whole list, paste it here, and see what is the outcome. Just to get an idea, okay, what is now this gene set? What do I have now at hand? And yeah, we don't get a lot of terms here, but for example, what is pretty fitting is actomyosin. I mean, with heart cells and the muscle cells inside, I guess it makes sense. Yeah.
Yeah, anyway, this has just got a 6 targets, right? So it must not be that GATA6 has, I mean, there's like a clear, you know, biological role annotated in gene ontology for this, but could be that individual genes are just important in that transition.
Exactly. What you could also do if you want to have a look at, or to make it more fast, so to say there's also a bash script which executes all the scripts I just showed you in one. And you can also hand it a list of transcription factors. For example, if you want to look at, I don't know, 5 to 10 different ones, just write it in quotation marks here and you will get a folder for each of them back. And then you can also pool, like, for example, all the GATA factors and then see, okay, what maybe is the outcome there.